My task this afternoon is to tell you a little bit about what FDA is doing in this arena. I suppose most of you, many of you at least, are pretty familiar with some of this. I'm going to focus mainly on the surveillance activities that are going on and tell you a little bit more about the program than about the data. The data are freely available on the web if you ever have been inkling to dig deep into it and look at different trends. I was going to sort of touch on that, but speak mostly about the program and changes to the program and the goals we're trying to meet by making some of these changes. I should point out Dr. Tom Chiller is on your agenda. He was going to present CDC activities in antimicrobial resistance. Tom is one of the lead epidemiologists on the meningitis problem and couldn't get away, I think, for that reason. So I've added a little bit to my talk, and Mary Torrance and I think we'll be able to tell a pretty complete story about NARMS. So in general, the Center for Veterinary Medicine has been dealing with this issue in concrete ways for at least a decade now or more. And the overall approach to it is to try to assess the relationships between use in food animals and the potential human health consequences. And this all came out of the original Public Health Action Plan that came out of an intergovernmental task force in '99. Where each of the different agencies proposed different ways to contribute to solutions to this problem. In fact, the public meeting on updating the interagency efforts is tomorrow in the Humphrey Building in D.C. So CVM's role in all of this is outlined here in this bullet list. It includes education, outreach activities, research activities. Many of you are familiar with the new revised safety assessment process and guidance for Industry 152, which adds human food safety and resistance as a component of that safety evaluation. Revised judicious use guidelines. This is guidance for Industry 209. I'm sure most of you are also very familiar with, and Dr. Carnival is going to speak more about these documents tomorrow. Along with 209 is industry guidance on meeting the requirements of 209 and guidance 213. Update on the Veterinary Feed Directive, enhanced surveillance activities. This is what where NARMS fits into this overall strategy. There's currently an advance notice for public rulemaking seeking comment on better ways to get antimicrobial use information. That's quite a challenge. And then also working internationally to at the very least, try to standardize methods and reporting approaches that allows different countries to make good comparisons about what's being sorry, I don't know. I don't even know how to do animation, so when it shows up, it's an accident. Challenges of doing integrated surveillance. These are not small. It's getting accurate information is expensive. Getting samples is expensive. It's labor intensive. It requires ahead of time some understanding of the burden of foodborne illness in a region or in a country so that you can prioritize pathogens to target. A sound sampling scheme along the food chain is critical. NARMS was built on pre-existing infrastructure when it began, and that comes with it some limitations, and I'll tell you how we're trying to address those. Cooperation and good communication between the different stakeholders. We've talked a lot about that at this meeting, and communication is an unending challenge. And then collaboration and information sharing is also critical. It's really best done in partnership because of the challenges that go with it. Of course, it requires political financial support and recognition that it's an investment worth making. An established process for review and enhancement. NARMS has been reviewed a couple times by the FDA Science Board, and some members of that committee are with us at this meeting. And based on some of those reviews, a flexibility to adjust the programs if new hazards emerge that need to be monitored. Understanding the implications of the data and how those data can be better understood with research, publishing the findings to different audiences in an understandable way, using the data to formulate sound public health policy, and there's often disagreement on that, as we're all well aware, and then the international aspect. So there's a lot to going into this type of surveillance, and we try to be mindful of each of these challenges and try to put on a culture of continuous improvement and trying to address any weaknesses or defects as we are able to, both to identify them and to garner resources to address them. The objectives of NARMS are to monitor trends in resistance in foodborne bacteria. It uses a three-pronged approach, if you will, looking in food animals, retail meats derived from them, and then cases of human clinical infection. To get this information out to people who can make good use of it, we talked a bit at the tables I've been in today about how we've often treated this as our only real critical control point, or at least our only actionable item. If we see a problem, what can we do? Usually all we know to do is, what do we do about use, because it's the one thing we have some handle on that we think we can control. It'd be nice to know if there are other critical control points that also might be valid interventions to limit or reverse resistances as we find them. But number 3, conduct research to better understand what we're observing in surveillance. I think this is fairly important. I think that though there is a sea change coming in what we used to call research, at least in terms of the surveillance system, and that is the advent of next generation whole genome sequencing, and I'll touch on that just briefly as well. But the overarching goal is to assist FDA making decisions related to the approval of safe and effective antimicrobial drugs for animals, keeping in mind that safety also includes the human food safety considerations. So this schematic shows, I hope in a useful way, how we do the integrated monitoring, if you will, getting isolates from the human population. Of course, this includes people that have felt ill enough to go visit their physician. Their physician took a culture, isolated a foodborne pathogen of interest. And then every 20th Salmonella gets sent to CDC from all 50 states, and a proportion of Campylobacter from the 10 FoodNet states get forwarded to CDC for the susceptibility testing. And so their data are contributed to the program reports. On the retail meat side, we have 11 states that are collecting 40 retail meats every month. That's pork chops, ground beef, ground turkey, and chicken breasts, 10 of each every month, isolating the foodborne pathogens that are indicated here. Let me see, do I have an arrow? So these little colored boxes here indicate the organisms that are being tested in each of the components. Oops. CDC had attempted to do E. coli and enterococcus, but weren't able to sustain that type of surveillance in healthy humans. It's quite a challenge to do that. Usually it's college kids who want a card to Walmart, and you kind of have a biased sample population. And it's really hard to keep it going for long periods of time. And then if you look on, so all 4 of the target organisms shown here are tested in the retail meat part. And then on the food animal side, this was really built, as I said, on pre-existing infrastructure, mainly the HACCP compliance samples FSIS collects on a regular basis. And those compliance samples were coming from all of the FSIS labs, and then and that was Salmonella from all the major commodity classes. And then to look at the other 3 organisms, they came only out of spent chicken carcasses in the eastern labs. So you can see it was sort of a quilt work of different data and sample sources that have been historically part of NARMS. And I indicate up here at the top that this part of it, the non-human part of it, is being revised currently to address some of the weaknesses. And then the data from all agencies are put into a single report that goes out on the web. The separate participating agencies have also published their own individual reports, and we're trying to get away from that and have a single report. The program began in 1996. It began in that year with just human isolates sent to CDC from 14 sites, and you can see from this diagram how that part of the program has evolved over time, so that in 2003 we had all 50 states along with 3 local health departments in New York, LA, and Houston. The FoodNet sites were used to both to get the Campylobacter isolates, as I pointed out, and also to set up the retail meat testing, and that was originally done at the same labs for both purposes, and this shows those sites that have been used from FoodNet. So it was a marriage of programs, both of which came out of Clinton's Food Safety Initiative. I've mentioned this. This shows the sources of the animal isolates and how Salmonella came from all the groups, or all the animal commodities from the 3 main FSIS labs, and how chicken carcasses were used for Campylobacter, E. coli, and enterococcus. And the E. coli and enterococcus are non-type specific organisms, or commensals that are there as sentinels of resistance. We're not trying to look for different serotypes of E. coli or doing any type of selective culturing. And there's some good reasons for doing that, but it does have limitations as well. So now we're going back a few years. In 2000, so I should say that there was a long-standing recognition that we needed to change some of the things in the program because our sampling wasn't as good as it could be, especially on the animal side, where we're getting all 4 organisms only from chicken carcasses. HACCP had become less random and more risk-based in its sampling, so that was biasing it even further. And so when we asked for the FDA Science Board to review NARMS in 2007, and Dr. Lonnie King was instrumental and chaired that committee. Larry Granger, who was here, was on that committee. Scott McEwen, Glenn Morris. I can't remember everybody exactly, but it was a very good committee that addressed 4 issues in the program that we presented, namely sampling, our strategy in research studies, evaluation of our plans for data harmonization and reporting, and were we doing the right types of analyses, were the reports digestible to a wide audience, and then the international activities and how much effort we should put into that and how we might play a leading role in some of those initiatives. So it was a very thorough and comprehensive analysis was done by the FDA Science Board at the time, and it's not my intention today to try to review all the recommendations and findings, but they did encourage the research platform and understood the value of that because you really can't sample everything, and sometimes you have to dig a little deeper to understand the limits of the inferences you can make from the data, and said that there should be more emphasis on hypothesis-driven and collaborative research. And this is what I was alluding to before. A lot of it had been, well, okay, we have these isolates of this serotype of Salmonella, these isolates, the same strains in food animals and retail meats in humans. In other words, are they making it through the food chain? Which really became sort of an exercise in genetic surveillance, by and large. It was looking for indicators of source, resistances to compare at the molecular or at the DNA sequence level. And so it was really just more of a refined monitoring type approach. They pointed out the need for an integrated database and to make reporting more timely, but anyway, to make reporting more timely was highly recommended because we were really lagging behind in the time it took us to get the data out, a couple of years originally. But we have been working to build the database and are still working on that. They strongly endorsed continuation, expansion of international activities, and part of that was building laboratory capacity in other countries for integrated surveillance. And by integrated, I mean from all 3 of the sectors that are included in NARMS. And then quite a bit was said about sampling and what we ought to try to do, and if we can't achieve true national representative and random sampling, that we might want to stratify the data in different ways. And we attempted after this, and you can read this report online if you're interested on the FDA website, but what we tried to do was sort of address each of these systematically and do things as we could, beginning with low-hanging fruit, as they say. And so after this report, we had a series of meetings looking at different aspects. The first to make sure that our own methods and approaches were harmonized within the program. And so we looked at, the microbiology being done in the lab. We set up groups to help communicate better and just talked about processes within the lab and came up with our own laboratory methods manual, which DOD has since picked up and is using in their enteric labs in different countries. Excuse me. The second time we met, a year later in Rockville, we talked more about the data management and warehousing activities, analytical tools, and what we thought would be best not only to make the data easier to analyze, but also to make the reports more timely and to make the data available on the web in a way that anybody could go and understand easily. And it was at that time that we started developing our strategic plan. Again, now in 2010, we focused mostly on the international partnerships, talking about countries, sort of a lessons learned type of approach of other programs that are similar to NARMS in other countries, different ways in which they overcame some of the challenges. And really, it's getting just the 3 sectors to communicate and work together is a huge challenge. And I've really seen this in the WHO work. The Ministry of Agriculture and the Ministry of Health often don't want anything to do with each other. And so that's been apparent in China, where they've got all the technical capacity, but they're not really all that interested yet in working together. On the research side, we sort of focused on that and talking about different things we ought to be trying to anticipate, especially as supply chains are changing so rapidly. And then last meeting, where we came together in a public forum, we talked more about the revised animal and retail meat sampling. We had a lot of stakeholders present at the meeting, and looked at different ways we might be able to get a better sampling design in place. And then this year we had another meeting, which I just note at the bottom. We had never had all the retail meat testing sites together in one place, and to try to understand each other better and how NARMS fit into their work, everyday work world, and learn from them some of the ways we could save resources and still achieve our goals. So that was a very productive meeting we had this year. So ultimately, the upshot of all of this, the Science Board review, and I should point out they came back in 2009 after this initial review, and we gave updates as part of a comprehensive review of FDA research at the time. But out of this came a 4-point strategic plan that, again, is thematically derived from the original review, where, and again, this is on the web if you'd like to look at it. But it talks about the sampling issue and the research and all the items that I've mentioned. Excuse me. In 2011, there's a couple of initiatives that we added to NARMS that hadn't been done in a systematic way before. At least some of them had. And one was to expand outbreak testing, or testing of outbreak isolates. There's a lot of effort right now in FDA and CDC and USDA to try to address the Institute of Medicine's mandate that we ought to, in approaching food safety, first get a really good understanding of the most significant microbial contaminants. And so part of that attribution approach is, can we use outbreak data, or isolates from outbreaks, to understand attribution just as well as we might from other types of surveillance? And so CDC has added outbreak isolate testing to their routine NARMS work to see how well those data will be generalizable in attribution models. Also, CDC has quite a few of programs that are in silos, as they say, and old legacy systems of software that don't talk well to each other. And we've been pushing them to try to link NARMS and PulseNet, something very simple. We do PFG on all the isolates we get from the other sources, but CDC has PulseNet over here and NARMS over here, and never the twain had met. And so we've really pushed them to try to link those databases, because it's obviously a lot of lost opportunity to understand the epidemiology of these diseases without that. So they've made effort starting in 2011 to try to address this as well. I'll skip to the bottom quickly. On the FDA side, for reasons I won't take time to go into now, we added 3 retail meat testing states, Missouri, Louisiana, and Washington, who are going to start full-time doing retail meat testing in January. And again, continue with the database development. The most material changes we've made recently, and this has been a tremendous challenge, and Mary Torrance is going to say more about an important part of this, is to try to overcome the limitations we knew existed in the animal component of NARMS. And we're taking a 2-pronged approach to this. One is to look at getting isolates on farm to have a true farm-to-fork surveillance system, and as part of this, to explore ways to get antimicrobial use information to add context to these samples. And I'll say a little bit more about that. And then also on the FSIS side, where we've been relying on HACCP samples, there's a lot of reasons that this is not the best sample set. And I could just say quickly that most of these are carcass swabs collected at the end of processing, which I would describe maybe more as retail meats that had not been cut up and shipped, and less like an animal sample. So these are isolates from sample sources that have been exposed to the environment of processing, to antimicrobial treatments that might be part of that processing. And so it was really hard to say this represents what was on farm at that point. So we tried to just move closer to the front door of the processing plant and on farm to have a better farm to fork data mining source. So I think that, well, this is a few more comments from the Science Board on the animal sampling about it being representative. On-farm data are essential. They recommended understanding the movement of resistance from farm to fork. So on-farm, there are two components, as I alluded to. One is to try to get isolates actually at the farm, and the other to do better implant sampling. So USDA ARS is leading pilot studies in beef and dairy and swine, turkeys and broilers to see how is it logistically feasible to do this type of pre-harvest sampling on a routine basis. Can it be done in a way that is random and representative, or at least quantifiable at some level as being representative of national production? And so it's using a randomized approach. It's collecting fecal samples, and we're developing a use questionnaire or a questionnaire to get use information. And again, the idea here, and there's sort of at least two approaches at FDA. One is the national sales data, and I know there's been a lot of conversations about this probably here and other places. National sales data by class, which is bulk annual sales by class, and it's not very granular, as they say, and trying to wet it to the resistance data. So one thinking is if we can get just use information to give context to that sample, and not with the intention of trying to extrapolate it to national. I'm not sure how much the national helps in this case, but just try to give some context to this sample to understand the selection pressures at some level in which the sample has been exposed. Then we do a better job, I think, of trying to understand the relationship between use and resistance. And that's another, good use information is another sort of long-standing recognized weakness of NARMS. The other animal part of NARMS is implant, as I mentioned. We've been working with FSIS, and they're going to collect samples for us in a randomized way that is nationally representative, and they're going to be testing cecal samples instead of carcass swabs at the end of the line. So we think this is going to be much better indication, and the data support that this is a much better indication of what's on farm, but I don't want to, we're going to recognize that it isn't on farm, right? So these are animals presented for slaughter that have been through transport and lairage, and so understanding, the limitations of using this as a way to signify what's on farm is something that we're mindful of as well. And I note in here that this will better reflect the consequences of vet antimicrobial use and less confounded by plant contamination, and that's not a trivial issue. You saw Randy Singer's slides of the E. coli plasmid that has Blossium Y. Right next to Blossium Y is a gene called SUG-E, which gives pretty high level MICs to acetylpyridinium chloride, which is a carcass wash. So you would expect that there are implant processes that would be skewing the resistance data you described from HACCP samples. It's not without value, but it's not the best design for the purposes of NARMS. So in the implant sampling, at the end of the day, we've finalized our agreement with FSIS. They're going to include or attempt to include at least initially all plant sizes. They're testing all the 4 bacteria I mentioned, Campylobacter, Salmonella, E. coli, and Enterococcus from the production classes indicated here. They intend to continue doing the susceptibility testing on the HACCP isolates. It is a rich source of isolates and has value. And so we're not surrendering anything, at least to date, but simply adding to our source of data. And then the goal is random and representative and sustainable. And sustainable is not a trivial issue. The work that we're trying to do on farm is a completely voluntary approach, and we don't want to be at risk of losing the animal component altogether if that voluntary approach, for whatever reason, didn't work out long term. So here's a quick comparison of the differences in the old and the new system. Basically, we'll have isolates from all sources, not just from spent chicken carcasses. This describes the retail meat aspects and how in 2012, that should say January 2013, really, for full-time testing, we'll have 14 sites altogether. Only 3 or 4 of the sites do the commensals, but all the sites do Salmonella and Campylobacter. And then we have a randomized approach. It's sort of it's randomized in the areas in which the labs can actually go and collect samples. And so this shows how the retail meat testing over the years, and well, if you look here, you can see it's expanded since 2002, where it started with 5 states. and now it's at 14, and you can see the number of samples tested each year has gone up, and next year will be nearly 7,000 annual samples. I mentioned I'd say something about the data. I mean, these are the Salmonella prevalence data from retail meats over the years. You know, it hasn't changed dramatically. If you look at the first and last years of this, they're pretty similar. The first level of information that we glean from this, you all are familiar that serotype often drives resistance, or at least there are restrictions, it seems, between serotypes for acquiring different resistances. And so the first level of data we see is if we compare the top serotypes in humans with the top serotypes from each of the commodities or the sources, we can get some idea of where resistance might be arising that is reaching it to clinical cases in humans. We have, you know, from this as well, the changes in these serotypes, and this shows the changes in the human serotypes. I think this is 2010, the top 4 in 2010, how they've changed over time. You can see in chicken breasts, the serotypes, you know, Kentucky's an interesting serotype. It's often the most common one in broiler sources. It doesn't cause much disease in humans. You know, I think there's something to be learned from that. But lately, Typhimurium, you can see, has started to supplant Kentucky. And it almost looks like there's temporal displacement of one serotype for another, and we see that in other sources as well. This shows, you know, I know this is quite busy, but this shows glom together all the resistances in the retail meat isolates over time. And in Salmonella, we're really concerned mostly about extended spectrum cephalosporins, which is ceftriaxone here, AXO, and quinolone resistance. But we've never seen Cipro resistance in Salmonella from any source, so that's good news. This shows how ceftriaxone resistance changes by serotype over time. Again, if we know this information and we know where these specific serotypes are coming from, it helps understand places where mitigations might have the most effect on human health. And this shows the same for poultry sources of ceftriaxone-resistant Salmonella. I just have a couple minutes, so I'm going to skip a little bit quickly here. Ceftriaxone resistance predominates in some serotypes. Here you can see it's Typhimurium, Newport, Heidelberg, but it gets around. In year to year, we find it in a wide variety of different serotypes. Here's the fluoroquinolone resistance in Campylobacter, and the black bar shows where the withdrawal of the approval for fluoroquinolones in poultry and jejuni and coli. Jejuni seems to have ticked up slightly, coli ticked down slightly. On the whole, if you combine them, it's pretty much just wobbling around the same rate it was when the withdrawal went into effect. This is something we've observed recently in Campylobacter coli, which is, we didn't see any gentamicin resistance to speak of for many years, and we've watched it really take off in the last few years, and we're trying to understand why that might be. Although I understand in 2012 it looks like it's on its way back down. So it'd be great to have use information that went with those samples, but we don't have that. I'm not going to spend time, I don't think, on this, but if you've been to our website online where you can see the data, there is an interactive tool here. Actually, the mouse isn't working, so I can't show it to you, but you can click on different sources and different drugs and look at line graphs over time. And I think I'm stuck. Sometimes that graph gets stuck. I think I have too many slides, but do I have a few extra minutes for Dr. Chiller's time or no? Okay. All of the isolates in NARMS go into PulseNet. Even though sometimes CDC is not sure which ones have, but as I mentioned, we're rectifying that. But we've submitted all ours to PulseNet. And while NARMS is not a disease monitoring system, it's really a post-approval safety monitoring system for food-animal antibiotics. That's how I describe it. But now we've got so many isolates in the program that it often, CDC and FSIS will look to it and when there's an outbreak and say, well, have we seen this before in NARMS, this serotype or this pulse field gel type? And sometimes it's directed outbreak investigations, including that Heidelberg outbreak in ground turkey in 2011. And this shows just the PFG data, or patterns that were associated with illness in the food products that could be tested, and then how this pattern had been present in NARMS for quite a while. Now, I think that the use of whole genome sequencing has been alluded to already. I really think we should all not underestimate the sweeping change this is going to bring to infectious disease epidemiology in the very near future. It's going to supplant, as I said, what we've always called research, PCR gene detection of resistance, comparing strain typing methods for discriminatory power. PulseNet itself will eventually be supplanted by this. And we showed in the Heidelberg outbreak that if you took these isolates with exactly the same PFG pattern, you could, through single nucleotide polymorphisms, distinguish the outbreak strain from the historical ones. And I'm going to skip over that and just summarize. So NARMS, we've been at it for quite a long time. It's a very good source of comprehensive susceptibility data. It has and can be used for regulatory decision making, including the pre-approval of new antibiotics in the 152 safety assessment. It's the most extensive national program for integrated laboratory-based surveillance of bacteria, I think not only in foods, but as far as receiving isolates, probably of any program. It has leveraged existing public health infrastructure and works well only as a collaboration between the three federal agencies. We are making improvements to it to overcome some of the weaknesses in the sampling design. It is a place where we have lab capacity and now an expanding team of people with lab capacity to look at different emerging hazards or hypothesis-driven research questions. The challenges, I've alluded to some of them right at the start, but we're really focused now on overcoming the sampling limitations, as I've said. But, you know, we have to have the flexibility to look at other things as needs arise. And so we've been asked not only by the Science Board, but others to be looking at seafood because we import so much of it. And how much resistance might we be importing? Is MRSA a foodborne hazard? And so on. So we are transitioning to whole genome sequencing. And we are looking for ways to get better drug use information. And the goal at the end of the day is to have data that are more reliable. So FDA is often accused of making decisions without complete scientific information. It's, it's a charge that's always possible to make. Hopefully by making the program more robust like this, we'll be less susceptible to those sorts of criticisms.